Great pleasure to welcome you at this very special, actually, seminar in the series of the Department of Epidemiology. This is so-called Works in Progress seminar, in which postdocs, others present what they are working on. And in this case, we even have to use an Americanism. We have a double header. And it is my great pleasure to introduce Laurie Chipnick, who will introduce the speakers then. Laurie is assistant professor in the Department of Epidemiology, biostatistician in Mass General Hospital, and in this case, that is why she sits here. She's director of the GINGER program, the Global Initiative for Neuropsychiatric Genetics Education in Research. GINGER, great name. Laurie, it's wonderful to have you, and I am very pleased to give you the floor to introduce our speakers of today.
Great, thank you so much, Bert, and thank you to the department for allowing us to do this. For those who do not know, the GINGER program has been running since 2017. We work with fellows at various sites across Africa, East and South Africa. These are fellows in collaboration with Kirsten Conan's NeuroGap project and Elise Robinson's NeuroDev project. I am very excited to be talking to you today, actually from Mexico City, where I had a bit of a Wi-Fi issue, so I apologize. So GINGER is called the Global Initiative for a reason, and it is very exciting that as I said, speaking to you from our sites in Mexico City, and being able to introduce two of our alumni fellows from one of the eastmost parts of Africa, Kilifi, Kenya, to one of the southern tip of Africa, Lerato Majara. So
we must come to the conclusion that Zoom is good for some things, at least, and in this case, it allows us to connect. Yes, thank you.
And I do like to say GINGER had been doing virtual classrooms before it became popular. But credit also to our wonderful Fran Cook, who has given us a lot of advice over the years as well. So without further ado, it is my utmost pleasure to introduce Dr. Lerato Majara, the very newly minted PhD Dr. Lerato Majara, with the ink still wet. So bravo for that. She will be talking about low generalizability of polygenic risk scores in African populations. I will say that GINGER brings together psychiatric genetics, so a genetic component and an epidemiologic component and a biostatistical component. Bert, would you like me to do both introductions now, or should I do the second one when Lerato is done?
I think that's better the second time.
Okay, yeah. Okay, thank you. Please take the floor, Lerato.
Thank you for the introduction, Laurie. I am very excited to be presenting here today over Zoom, over 2 different time zones. And as Laurie said, my presentation is about polygenic risk scores and their transferability in African populations. And this is a project that I was involved with as a GINGER Fellow. And this project was mentored by Alicia Martin, who is based at the Broad Institute. So because we're talking about polygenic risk scores, PRSs, I'll start by giving a definition. So they are a way to quantify an individual's risk of potentially developing a disorder or a specific phenotype, and they're calculated as the sum of allele effects weighted by their effect sizes. And the way that works is you first conduct a genome-wide association study, a GWAS in a discovery sample. Then you select SNPs from and above SNPs from the GWAS itself. Then you compute the PRS and you apply the PRS to a target sample completely independent of the discovery. And then you assess how the prediction accuracy of this polygenic risk score is in identifying or predicting the phenotype in the target population. And this can be done within the same disorder across disorder and as well as between disorder types. So this cartoon here is an illustration of how polygenic risk scores are calculated. So in this example here, the beta of the CLO, the affected allele here is negative 0.2. So then the scores will be calculated as the sum of all the effect alleles there, weighted by all of the effect sizes. So in this example, the individual's polygenic risk score would be 0.052. So the first application of PRSs was in humans was in 2019 in this landmark study by the International Schizophrenia Consortium, where they assessed the prediction accuracy of European ancestry-derived polygenic risk scores who with schizophrenia in different cohorts, as well as in cohorts with bipolar disorder. And they included here non-psychotic phenotypes as sort of a negative control. They used the pruning and thresholding method. So the different shades of green here indicate the different p-value thresholds that were implemented. So the findings here were that prediction accuracy was highest in the European ancestry populations, as indicated by these large bars here for this MGS cohort, as well as the cohort in the O'Donovan study, which is also primarily European ancestry individuals. They also showed that you're able to predict bipolar disorder in 2 different cohorts. One of the most notable things from this publication was the very low prediction accuracy here in African Americans. So you do well at predicting schizophrenia within the same ancestry and across disorders, but not within the same disorder across ancestry. So since this first application, there are now 247 scores that have been computed across 218 traits according to this PRS catalog. So this was in 2009. So forward to 2019, 10 years later, the same sort of phenomenon is recapitulated. And I'm showing here a figure from Alicia Walton's paper where PRS accuracy was computed for non-psychotic phenotypes. And again, they're showing that prediction accuracy is highest in European ancestry individuals if the discovery was European ancestry. And it declines by 1.6 times in American and South Asian population, and 2 times in West Asians, and 4.5 times in Africans. So essentially showing that prediction accuracy decays with increasing genetic distance from the discovery data set. So we know that Africans are not a homogeneous group. So the prediction accuracy, or the extent to which the accuracy varies within African populations, hasn't been extensively investigated. So the aim of the project here was to look at the generalizability of PRS within and among diverse African populations. So 3 fellows were part of this project, including myself. The other fellow is Nastasia Kuhn, who is a senior lecturer at the University of Cape Town here in South Africa. The other is Alan Klimke, who is a postdoctoral fellow in Neurogap, and he's based at Makerere University in Uganda. So the project was done in 3 parts. The first part involved a simulation using the African Genome Variation Project dataset, where we were investigating transferability of polygenic risk scores within African populations, where the discovery and target datasets were Africans. The second, in the second part, we used the Drakenstein Child Health Study at CHS, which is a birth cohort study here in South Africa, where mothers are recruited into the study during their second trimester and followed up. And this data set comprised individuals who self-identified as Black, as well as those who are mixed. So here we used European ancestry hallucinogenic risk scores and assessed their predictive accuracy in the South African population. Lastly, we assessed European ancestry derived hallucinogenic risk scores in the Uganda general population. So for the purpose of time, I'll only be presenting findings from this last part. So as I've mentioned, the aim here was to assess predictive accuracy of European ancestry-derived hallucinogenic scores in the Uganda general population for the GPC, which comprises 4,778 individuals from 9 ethno-linguistic groups in Uganda. So for this part, we assessed 34 anthropometric traits, including blood factor, blood pressure, glycemic control, liquid and liver function traits. We used 2 sets of data sets. The first was individual level genotype data. So we used whole genome sequence data from GPC, as well as data from the UK Biobank, comprising over 350,000 individuals living in the United Kingdom. The second part, or the second set, was GWAS survey statistics from GPC, UK Biobank, and Biobank Japan, comprising East Asian individuals, as well as PAGE, which largely comprises African American and Hispanic and Latino participants. I hope you can still hear me clearly. It's raining here and we have a sync. I'm struggling to hear myself, so I hope the audio is still okay on your side. Okay, so we assessed the predictive accuracy of European ancestry-derived PRSs for the phenotypes in GPC. And in general, there was low prediction accuracy for all of the phenotypes. The highest was MPV, which is mean platelet volume, with a maximum R squared of 0.038. Even though this is the highest here, it was still very low because predicted accuracy in European individuals in UK Biobank for the same phenotype is 0.22. So obviously several folds higher than what we see in the Ugandan cohort. That's the case for heights as well. We went on to look at predictive accuracy across continental ancestries as well as within African ancestries. And this was done by withholding 10,000 individuals from UK Biobank with European ancestry and using those as the discovery data set. And then we computed polygenic risk scores for the different continental ancestries. So this here is similar to the slide that I showed in the introduction, where polygenic scores are highest in the European and decline with increasing distance between discovery and target. And again, accuracy is the lowest in the African group. We went on to further divide the African group into different geographical regions. And there we saw variable prediction accuracy between these groups. Accuracy was highest in the Ethiopian group and was reduced in the Admixed populations, East African population, West African, and was the lowest in the South African populations. And this observation tracks with population history. So the Ethiopians have a closer genetic relationship to Europeans, and hence why prediction accuracy, you'd expect that prediction accuracy would be higher there, where the South Africans are the most divergent from the European ancestry population, and hence the lowest predictive accuracy. Finally, we compared predictive accuracy between the East Africans living in the United Kingdom, so those that are part of the UK Biobank. We compared that to East Africans living in Africa and Uganda. And we're seeing higher prediction accuracy in the East Africans living in the United Kingdom. This difference could be attributed to the difference in the environmental factors between these two groups. So there was a larger difference in prediction accuracy between ancestries than between. Next, we compared the predictive accuracy of single discovery cohort to a multiple single ancestry cohorts to multiple ancestry cohorts by including samples from Biobank Japan, comprising 162,000 individuals, as well as Page with 50,000 individuals. So the multi-ancestry comprised all these three cohorts, and that was compared to UK Biobank below. In general, there was an improvement in prediction accuracy across phenotypes, but particularly for the groups. Sorry, so predicted accuracy here is shown by the population groups. AMR is American, MID is Middle Eastern, CSA is the Central and South Asians, and the others are still as they were before. So prediction accuracy was improved in the American group, and we're seeing, so where there is an annotation, of the phenotype, there was a disproportionate improvement in prediction accuracy. So in the American group, that was in the mean muscular hemoglobin concentration. We also saw improved accuracy in the East Asians and in the African group as well. So this improvement is reflective of the ancestries that were included in the discovery cohort. For the African group, we saw major gains in prediction accuracy for mean muscular hemoglobin concentration, as well as the blood cell count. We then went on to look at what was driving this major gain for these two phenotypes, specifically for the African group. So we plotted the p-values for the single ancestry compared to the multi-ancestry discovery datasets against each other. And we annotated the SNPs that were deviant from the diagonal line. And we observed that it was the population enriched variants that were driving this major gain in PRS accuracy. So for example, variants in HBS1L have been associated with sickle cell and beta thalassemia, phenotype endemic to African continent. Similarly, for WBC, it was the variants in these DOC, ACKR1, that were driving this enrichment, this gain in accuracy, and those have been associated with malaria resistance, which we also need to be talking here. So in summary, I showed that there's low generalizability of polygenic risk scores within African populations due to two things. The first is that there's a high African diversity. Here I showed variable prediction accuracy between African populations. The other factor was the environment, where I showed the difference in prediction accuracy between the East Africans living in the United Kingdom compared to those living in East Africa. There's been a lot of conversation about, and also a lot of encouragement of the use of polygenic risk scores in clinic currently. But so this, I think, I put this in quotes because it's the title of the Alicia Martin's paper, saying that the use of current polygenic risk scores may exacerbate health disparities. And as I've shown here, if we were to use European ancestry-derived GWASs, because that's the most GWASs are from European ancestry populations, then PRSs wouldn't do very well in predicting these phenotypes in African populations. So that would potentially exacerbate the health disparity. So I'm saying all of that to say that there's a critical need for large-scale studies in African populations that represent, to a large extent, the diversity of the African continent. And I think that would help in sort of getting rid of the Eurocentric bias in GWAS studies in terms of the samples that we're using for research. I think that's also important, is that diversity is not only represented in GWAS samples, in samples, but also with the researchers doing this kind of work. So we need to really make huge efforts in building capacity and training. And I think that kind of speaks to the work that Ginger is doing. And I've been very fortunate to have been part of such a program, and I still play a very active role therein. So these are my acknowledgements and my final slide. So I'll take questions, and also very curious to hear what people's thoughts are about including polygenic risk scores in the clinic today. Thank you very much.
So yeah, we're moving from the southern tip of Africa to the east coast of Africa. And it's my pleasure to introduce Dr. Simon Kariuki, who's going to talk about can exposure to infection determine outcomes or phenotypes in epilepsy? So for those who are not interested in genetics, this is for you. We're going to the other side of our neuropsych in neuropsych genetics. So take it away, Simon.
Thank you very much, Laurie. So today I'll be talking on the impact of epilepsy, infections in epilepsy beyond them causing epilepsy, because I know many people have really concentrated on the initial phase of them causing the disorder. And I just wanted to start by saying that I come from the KEMRI Wellcome Trust Research Program. Many know that we are based at the Kilifi, Kenya, but we also have other centers in Nairobi, as well as in other parts of Africa, including Uganda, with so many other collaborating projects across the many other parts of the world. And I wanted to start by demonstrating to you that infections that have a propensity for the brain usually are very, they have a special preference to the developing period of the brain, and that is the first 5 years. And if you can look at that figure, you can see that most of the functions in the brain are developing in the first 5 years. Myelination, visual cortex, seeing, and you can see that by the time you are 15 years, when most of the functions have developed, if you had any exposure to infection, then you'll have sustained a lot of damages that you are going to live with for most part of your life. And here, when we are talking about infections, we are interested in the ones that are very common in our settings here, starting with the parasites such as malaria, viruses, and also bacteria. And I'll be giving you an outline of the many neurological involvement that these disorders can bring about. And what usually happens is when you get an infection, in addition to getting the systemic disease, some of the infections have a propensity for the brain, and they are able to reach the brain directly, either through the epithelium of the blood-brain barrier or through the cerebrospinal fluid colloid plexus, and sometimes through the neurons or the axons of the neurons on the surface of the skin. And I'm just outlining there the way the bugs have become very clever, that they can be able to cross the blood-brain barrier through several mechanisms, either in between the cells or through the cells in the process of transcytosis, or sometimes fooling around with the existing receptors so that they can be able to get inside the brain. And the mechanisms for brain damage when these infections get in the brain are not any different from what we see for other neurodegenerative disorders, So you have an activation of microglia, then you'll also have activated leukocytes. That's the white blood cells that are resident within the brain, and also some reactive arthrocytes. So those happens to damage the brain, and neurotransmission there is affected. And when that is problematic, that's when you can start getting problems such as seizures and epilepsy. So those infections with a propensity for the brain, for example, for bacteria, is the disease called meningitis, viruses, you have meningoencephalitis, parasites such as malaria. We do talk about a neurological involvement, which some people know as cerebral malaria, and I'm providing the estimate there in terms of incidence. And you can see it's quite very high, and it cannot be ignored. But they do lead to a lot of neurobehavioral manifestations. I've listed quite a number of them there, ranging from intellectual disability to mental health problems. But my focus today is mostly going to be on seizure disorders, specifically epilepsy, which is usually defined as a history of 2 unprovoked seizures. And the reason why we focus on epilepsy, it's because it is very common, especially in low and middle income countries. And this is usually thought to be related to the high incidence of infections that I'm providing on the right panel there. For example, Toxocala cani, Falciparum malaria, Toxoplasma gondii, as well as Onchocerca volvulus. And what usually happens is these infections, particularly those that are able to reach the brain, they can be able to cause localized damage. But there are also others which do not reach the brain, but they can be able to affect the immune system, resulting in inflammatory cytokines that can indirectly damage the brain. But the examples that are given there are largely based on the case control studies. And you know, the validity of case control studies just provides association that may not be able to give us a lot of information concerning the causality. And therefore, if you want to increase the validity to infer causality, you need to consider other options, for example, the cohort studies and the intervention studies. But if you look very closely on the x-axis, that's very costly and sometimes may not be very feasible in many low and middle income countries. However, there are still a few cohort studies that have been conducted, especially in our settings here, and they are focused at specific disease states. So the first one are cohort studies that are supposed to establish whether infections can subsequently lead to a downstream development of epilepsy. I've given an example there, which yielded an odds ratio of 4.4. And then we've also been able to look at the process of epileptogenesis using longitudinal studies through a paper that we published in 2016. There, we were able to establish that if you get acute symptomatic seizures early on in the infection, then you are at an increased risk of developing epilepsy in the long term. What we've not been able to do, though, is to examine what happens when you already have epilepsy, you are living in an endemic area and you continue to be exposed to these infections. So I wasn't aware of any study that has looked at that, and therefore we realized that there is a very big research gap there that needs to be addressed. So is it important to investigate or to examine the load of infections when somebody already has epilepsy? I think it is very important because we know that epilepsy usually starts at different time points, and sometimes because infections are very common in early childhood, that might determine the time at which it is going to start, and therefore the duration that you are going to live with it. And we also know that some of the features in epilepsy, particularly those which usually affect one part of the brain, are usually related to the infections. But this has never been examined really, whether it is specifically due to the infections or it's an inherent characteristic of the epilepsy. So we are also aware that if you get acute infections, you also tend to have that propensity for very prolonged seizures and also multiple seizures. And again, we'd really like to look within the epilepsy group to see whether that is usually related to continued exposure to infections or not. So this led to this particular study, which was part of a multi-site study conducted in 5 African countries. and we were able to perform assays for 6 infections there, Plasmodium falciparum malaria, HIV, Toxoplasma gondii, Onchocerca volvulus, Taenia solium, and also Toxocala cani. You can see that most of them are parasitic infections, but we also additionally assayed for HIV because we know it increases the risk for development of those conditions. You can see some of them were not assayed in South Africa and also in Kilifi, Kenya, specifically on Onchocerca volvulus and Taenia solium, because they are not very endemic in that area. So we were able to use samples from unexposed adults to be able to develop the cutoffs that would be able to give us a positivity for these infections by simply taking the mean of the antibody levels and then adding 3 standard deviations, and anyone with antibody levels beyond that was considered to be seropositive. And now on to our results. You can see that if we stratify our data set by infection status, infection being the blue bars, the red bars, and then an infected population being the blue bars, you can see that most of the severe features of epilepsy, and this is within the group of people living with epilepsy. For example, focal seizures, status epilepticus, which is a very serious emergency, neurological deficits, intellectual disability, as well as having an abnormal electroencephalography, they were significantly increased in those with infections than in those without. But the biggest proportion of infections was actually found in those that were not using anti-seizure medications. So we tried to construct mixed effects model to see the direction which these features were associated with exposure to infections. And you can see there in the adjusted models, we found a number of significant features that were associated with exposure to infections when you are continuously exposed to them. Use of anti-seizure medication was on the protective side, but not using anti-seizure medication was increasing the risk, and so was status epilepticus with the greatest risk there at a risk ratio of 6.02, frequent seizures and cognitive impairments, which is also known as intellectual disability. And that was for all participants combined. And almost similar findings were also found for children, finding the same features being more reported in those exposed, continuously exposed to infections. Again, failure to use anti-seizure medication, convulsive status epilepticus, intellectual disability, and neurological deficits. Then we focused on the adults only. We found the same features that we had found in the other age groups, obviously with the exception of a few. But here the most important ones were frequent seizures and intellectual disability, but unlike children, we didn't find any association with the status epilepticus and the neurological deficits. So that was when you consider the presence of any infection. But if we look at each individual infection at the time, you can see that Plasmodium falciparum malaria in children was mostly related to anti-seizure medication use, but in adults, it was increasing the risk for intellectual disability. But the most important infection there is Onchocerca volvulus, which you can see is very epileptogenic because it's associated not only with the focal seizures, but also with the frequent seizures, status epilepticus, and neurological deficits. And we've recently come to find out that it can easily be prevented through mass chemoprophylaxis with the ivermectin. So we also tried to examine whether there was any interaction between the infections. And indeed, we found that you have both synergistic and antagonistic interactions with regard to the risk for the severe features of epilepsy. So giving an example for falciparum malaria and HIV, where there is an antagonistic interaction there for status epilepticus, but majority of them, for example, Onchocerca volvulus and Toxoplasma gondii, interacted synergistically to increase the risk for status epilepticus. So we can make a few conclusions from this work and conclude that a continuous exposure to infections based on these findings is associated with the severe features of phenotypes of epilepsy. But we can see some differences between children and adults. status epilepticus being the most important consequence of exposure to infections in children with epilepsy, while frequent seizures were useful in adults. But we can also see that these features are also dependent on the type of infection that you have, probably underlining the fact that these infections have a different pathogenetic mechanisms and most likely also epileptogenic processes. So we are also seeing that infections sometimes can interact to lead to these features, and the direction can be either way. So they will not only increase the risk when they interact, but sometimes when they interact, the risk for that poor outcome can also be minimized. And then we also had findings concerning population attributable fractions, for which we found that 40% of the severe features of epilepsy were actually explained by the exposure to infections, clearly meaning that if we had interventions for reducing exposure to infection, then we are greatly going to improve the outcomes of people with epilepsy, given that we were actually also accounting for anti-seizure medication use. So therefore, I think the next step from there is to think about the interventions that can be set up in low and middle income countries, especially to prevent infections in people with epilepsy, improve outcomes, or even to prevent infections so that you don't develop epilepsy in the first place. We've done quite a number of studies in the past, which we can be able to follow up in the long term to try to examine the first objective. And at the moment, we are concentrating on vaccination with RTSS, which had happened between 2007 and 2011. And we're just interested to see whether those children who are vaccinated with RTSS versus those who had not been vaccinated, do they have improved neurobehavioral outcomes in terms of seizures and other mental health problems? So that would just be one aspect, but in the future, when we get an opportunity, we'll also be looking at the role of chemoprophylaxis as well as insecticide-treated bed nets. So we'll be able to provide you those results if we get an opportunity to speak here. And I would like to conclude by thanking everybody who contributed to this work, particularly the investigators listed here who contributed to this particular paper, which has just been submitted and is undergoing peer review. Thank you very much.